I'm Seda Kurt from the International Biogeography Society. Welcome to the fun lecture series on biogeography. Today, we welcome Joaquín Hortal, whose talk is titled Mapping Ignorance on Biodiversity Towards a Fair Account of Data-Driven Uncertainty in Biogeographical Analysis. While we wait for more people to show up, I'll go over a few slides about the upcoming IBS conference. Then David will introduce Joaquín and start the session. So the next IBS Biennial Conference will be held in Prague in January 2024. You can see the key dates on the slide and remember to register early to receive a discount. The conference website has all the necessary information, but if you still have any questions or comments, you can send an email or DM through the IBS social media accounts. And just a quick reminder that Joaquín's talk will be followed by a Q&A session. You can type comments on the chat, post questions in the Q&A section, or raise your hand to ask a question using the icons at the bottom of the Zoom window. So thanks for joining us today. And I think we can get it, David.
Thanks, Sarah. Thank you to everyone for being here today. It's a pleasure to see many people here as attendees. It's also a great pleasure, also for many different reasons. It's an honor to have Joaquín Hortal here today. Thanks, Joaquín, for being here with us. Joaquín Hortal is a scientist based at the Natural History Museum of Spain, but he has been visiting different countries during his time. He has been working in UK, Denmark, and Brazil, as far as I know, and if I'm wrong, Joaquín will tell us. The research of Joaquín spans from biogeography to community ecology, but it also spans from the area of fieldwork to models and simulation. So Joaquín is a really complete scientist, or a biogeographer in this case, that is able to link many different aspects of research to both answer basic questions, but also to gain applied knowledge. And I will not take more time from Joaquín. I will say thanks again to everyone for being here. Joaquín, the stage is yours.
Thanks, David, and thanks a lot for inviting me for this. It's an honor to be lecturing here for the IBS, for my primary society, and also it's an honor to do it in memory of Vicky Funk. And well, what I'm going to show today, it's sorry, I'm going to show my screen. Yeah, it's part of the work I've been developing for the last 25 years, together with many people. I'll share the acknowledgements later, which is basically about mapping ignorance on biodiversity and how we can account for uncertainty, as you see in the title. So one of the things when you start working on, sorry, it's not, the slide is not working. No. One of the things that happen when you start working with the biodiversity is you realize, I particularly like this, the title of this book from Kevin Gaston, The Biology of Numbers and Difference, is that biodiversity is about describing a lot of things and a lot of different things, as we see here, with different forms, functions, ways of living, etc. But when you start working on biodiversity more seriously, what you see is that we know a lot of species, but for many groups, we lack a lot of knowledge out of them, okay? And perhaps because I'm an entomologist in origin, I work with other groups, but I do work, my training is as an entomologist. I'm well aware that we know very little of invertebrates, at least compared with other groups that are better known, like chordates or plants. No, I mean, if we talk about fungi or viruses, etc., the picture is even worse. And actually, what we want to describe is not only a lot of species. It's the whole tree of life of species that are species and populations and individuals that are interrelated between them. They have distributions, as you see in the map below, and they interact between them in complex ecological networks and perform and interact in these networks depending on the many functional traits they have, which vary individually. So actually, it's a lot of information. As is something I usually joke with my friends who are scientists but are not biologists, is that our science is the science of complexity. Okay, so knowing that we know little about that, and we have to describe a really complex phenomenon, I think we have to go back a bit to the philosophy of science and think about what is scientific ignorance. I mean, recognize, as Maswell said, that we are ignorant about biodiversity. There are a lot of things, but we know a lot of things, but there are a lot of things that we do not know. And try to gain this conscious ignorance that Maswell said that is the prelude to the real advance in science, okay? And I particularly like the way Stuart Firestein, he's a neurobiologist, pictures it in this book I'm picturing there, and
But
which is like, sorry, yeah, which is like that we are when we are working on a particular field. It's like when we throw a stone to a pond and it starts to create waves that expand. That's the same as when we start to create knowledge and accumulate knowledge on a given field. And the more we know, the larger those waves get, and the more we realize we don't know. The whole universe we want to know is the whole size of the pond. In our case is biodiversity, okay? And the more we know, the larger is the border, okay? And what Maswell said and Firestein develops is that if we are able to describe the borders of that knowledge and how are those borders relate to the unknown, to the things that are nearby, we are going to find several areas that are like interdigitations or things, something like that, where we have accumulated knowledge in particular parts, but we have small gaps or biases in the in-between, okay? And it will be quite efficient for us to identify those gaps and fill them. Just to give you a short example coming from another field, the Higgs boson was described that way. The Higgs boson was identified as a particle that should be that way if the rest of the particle physics was right. They identified the characteristics of an unknown particle. And after several decades of working on it, because it's a different field from ours, they did find Higgs boson with those characteristics. So how does it work for biogeography and biodiversity? I particularly like the way that Rob Caldwell and John Connington put it in this paper from several decades ago, that the information the universe we want to describe, that pond, the size of that pond, is what will be contained in a magnificent and omniscient GIS, which contains information on all the Earth's living species with the capacity to display any level of the Linnean hierarchy on any spatial scale for any season of the year. If we had those data, we don't need statistics. We just extract the data we want, and then we plot it, and we take the exact response to the hypothesis we want to test, okay? That's not how things work, and it's completely impossible to obtain this full and omniscient GIS with the whole knowledge. But I mean, it is good as a metaphor to realize what we do need to approach to, okay? Which is our final objective, which is of course impossible to reach, but like all good objectives, is always utopian, okay? It's good to know that we are addressing that way. And the tools we have for that right now are biodiversity databases. We're integrating a lot of information on the distributions and characteristics of biodiversity. You may know some of those databases. They are getting together. There are several more, okay? those databases contain millions of records on species, individuals, populations, genomes, etc., etc. But if we work on those with those databases, we realize that they fall short in describing biodiversity. And in this, we were not the first to describe some of the shortfalls of biodiversity. We just compiled them several years ago. And we compiled 7. We proposed some of them. Then another one has appeared, 8 so far. But basically, there are shortfalls in any meaningful aspect of biodiversity, okay? The Linnean shortfall is about the species we know, and then we've got the Haeckellian shortfall. It was recently described by Luis Faria and colleagues, which is about the different life forms. We don't know the life forms for many species, even we have described them. And then there are others. The one that matters the most for biogeographers perhaps is the Wallacean, which is the distribution, is the lack of knowledge about the distribution of a species, which was described by Brown and Lomolino several years ago. But basically, we don't know, the data we've got falls short for describing the responses of the species to the environment, the traits and their functions, the evolution, the dynamics of their populations, etc. You've got there that information. What I'm going to show here, rather than delving on those, on thinking about, okay, we don't know many things, is where should we go from there, okay? And I think we have to do at least 3 different things with this, now that we realize the problems of the limitations of the data we've got. We have to assess the quality of the data to make sure that the data we've got is good. We have to map the coverage of knowledge. This is similar to what Firestein was proposing, map that knowledge. And then we can also estimate the uncertainty in our models or our hypothesis testing or our analysis that is driven by the data, okay, by those limitations of the data. So starting with the first, with data quality, I'm quite happy to show, or I love to show a paper from Vicky that I was lucky enough so as to edit with Víctor García Milagros, and where they were showing the gains that we got if we really work in the data we are using or gathering from databases or from collection. In this case, it's one plant. They put several examples, but if you looked here, I don't know if you can see here my pointer, this white spot is where the location that was given for the plant in the record. And when they looked into the information in the paper describing that voucher, the plant was located there. Okay, this is at Table Mountain at Tepuy in the Guianas, I think, and or Venezuela, sorry. And this small uncertainty in the spatial location gives us a difference of several hundred meters in altitude. Okay, so if you don't check the data, you may get many wrong answers. Okay, this will help you to correct and filter out poor quality or unfit data when those data is quite important. Of course, I'm not saying to do that with thousands of records, but we can work out for a particularly important ones, okay? Then there is another aspect that we often pass away. I particularly like this paper by Losier and colleagues about the taxonomy, the quality of taxonomic identifications. This is in this paper, what they did is what we usually do when we do the species distribution models, that is go to a database online or to a couple of databases online, download the data about the species, make a species distribution model. Okay, this is the potential distribution or where we could find the species. But they did it for the Sasquatch, for the Bigfoot, which is obviously not a true species. And they gathered from 2 databases, one from footprints of the species and the other from people who had heard shoutings of the Bigfoot, okay? What they did was to compare it with the niche of the black bear, and surprise, surprise, it was exactly the same, okay? So there was a lot of misidentifications. There were people thinking that what they were seeing or listening to were Bigfoots instead of black bears, a real species. Okay, when we go to the real world, we have to be aware that many of the records we've got, especially now that we use data science, citizen science, is that some of those records will be validated by people who are really expert in the group and are going to identify the species with precision. This may not be a problem for some vertebrates, but it's really a problem. It can be for many birds and truly for many invertebrates and plants. And depending on the level of knowledge on who was responsible, the quality of that record will be higher or lower in terms of identification. In particular, if we use artificial intelligence to identify species like in some places iNaturalist is able to do right now, okay? So I think we can identify that quality and tag the records, flag the records with this information. We have been working in my group, thanks to Cristina Ronquillo and Juliana Strop, and this is currently under review. So we're saying please don't tweet, but this is going to be online, so doesn't matter actually. But we have developed a Shiny app which will guide you through all the steps needed. We have made a revision, but instead of making a review paper, what Cristina did was to put it all together as a Shiny app, which will guide you through the different steps of correcting and filtering out poor quality data or data that is not fit to the question you want to answer, and identify that and standardize, showing you the different methodological alternatives. As I told you, this is a revision paper, has a review behind the app, okay? So it will show you the different alternatives, and at the end, it will provide you with a report that guides you through all the filtering steps you need to do and the tools that you have to do that. This will help you make sure that the paper or the analysis you make for the paper or for the report or whatever fit really or have the necessary quality for the question at hand, okay? Other things that, sorry for that, yeah. Other things that is also necessary to do is the second step I mentioned, is mapping the coverage of knowledge. This is a paper from Juliana Strop, where we plotted the distribution. We mapped the distribution of GBIF records on seed plant species in Africa. In the upper right map, you can see the places with high completeness as red dots, which shows you that well-known places are concentrated in a handful of areas throughout Africa. But if you look for the left side map, we can also see that, for example, South Africa, which is the country with the better information, is also played with old information, okay? So we've got good geographical coverage for South Africa, but we've got poorer temporal coverage if we want to recover the current distribution of biodiversity. This identifies a gap. We need, if you are in South Africa, you need to repeat surveys to see if the old data you've got still holds up, or you need to, or the distribution of some species have changed, okay? This is quite useful to identify gaps in knowledge. We have been developing the techniques for this. Is a paper from Tadeu Sobral Sousa from Brazil, currently in Brazil, Tadeu, which is about the coverage, about describing the coverage of knowledge for, in this case, for fruit feeding butterflies in the Atlantic forest. And in this case, we wanted to map the coverage that we had about the distribution of those species, but also about their responses to climate, which is what you have in the right, and also with landscape variations, okay? We did not only map the total coverage, but also the coverage of rare environments and rare types of fragments in the landscape. the main result of this paper was that we may have a decent coverage of some of the main environmental gradients, not for all, but we have a very poor coverage of the landscape, the land transformation gradient. We have good data for small fragments and for big fragments, but not for the in-between. So we are not able to identify at which point, or which is the shortfall, sorry, not the shortfall, which is the threshold at which the communities of butterflies, of fruit-feeding butterflies, start to collapse. Okay, we lack this kind of data. So we need to gather data of fragments of intermediate size, if they are available somewhere, because as you may know, the only remnants in the Atlantic forests are either big or really small. Okay, but we identified that we cannot find that threshold. This kind of analysis can be used to identify new areas for survey. In the upper part, and these two papers came almost together the same year. One from this comes out of my PhD thesis, where we identified areas with high completeness, and we mapped not only the coverage, they had the geographical coverage, but also the climatic coverage, and identified which areas were better to survey to complete that coverage. And at the same time, Vicky was working with that, with Simon Ferrier and other people in the Guianas, Vicky Funk, and did that for plants and several other groups, identifying the areas, the areas that were well covered and those that were in need of survey. Okay, so one of the first utilities of this kind of techniques is identifying where to go and direct new surveys. You can extend that to other aspects of biodiversity. We have been talking about distribution, but in this case it's populations. Okay, it's the population dynamics. In this case it's with Javi Nori and other colleagues from Argentina. What we have mapped is the coverage we've got about the effects of land transformation on amphibians and reptiles worldwide, on all amphibian and reptile species, thanks to data from Mexico here. And well, the first result is that we don't have time series. We don't have any time series. Well, we have very little time series so as to provide any coverage of the temporal reactions of species and the population dynamics after land transformation. So what we had to do is to make a space by time substitution and identify places where we have native fragments of native populations from fragments of native habitat and populations from fragments with transformed habitat. Okay, and this is the map you're showing, and as you see, we see different percentages of unknown species. We see big, huge gaps in Africa and for reptiles in Asia, and strikingly, we see that, for example, for amphibians, we lack data in Europe. Okay, this is perhaps because in Europe, land transformation occurred a while ago, while for this same group, in South America, we've got better information because land transformation is happening right now. Okay, but this helps identifying us the places where we need more data to assess the dynamics of the population dynamics after land transformation in these two groups. We can also tailor this kind of knowledge coverage assessment. This is an example we can tailor it to particular questions. This is an example from Tiago Freitas' PhD, also in Brazil, and he was working with a family of catfish with Autenitheridae, and he wanted to assess how did the trophic niche evolve in this family. Okay, so we had to look at distribution to see if there were distributional patterns, of course, how complete is the inventory of the species, but also about he also needed to know how stable is the taxonomy, how stable are the phylogenies, and how good and how how good is the knowledge on the feeding habits of those fishes, and how is it distributed along the different groups. And this is what you see. Okay, I'll save you the details, but basically out of the 126 known species, only 47 were broadly covered within the good phylogenies and good knowledge on trophic habits, etc. Okay, so we can tailor those analyses for the questions at hand. Okay, so this covers the first two strategies I've mentioned, and then I'll go to the third one, which is the one we've been working for the last years, and I also presented as a first example in the IBS in Bayreuth, I think, but we have continued working on it, which is the maps of ignorance. It comes out of the idea of Sandbox, an American geographer, of creating atlas of ignorance that depict not only together with the maps of a particular geographical aspect, also the level of knowledge we've got about that particular aspect. For example, in this case, this map is the first map of ignorance, is about soil. The areas in the five, the white is areas without soil, but the darker areas are areas with no knowledge at the time. Is 1950, no knowledge about the soil, or progressively better knowledge about different soil characteristics. Okay, this is quite useful to show us how good or how uncertain is the information that we've got for each particular place. In the case of biogeography, our proposal in a paper by Duccio Rocchini and other colleagues and by Richard Leidel and myself is that we have to look at different factors, but at least three that may be compromising the quality of our knowledge of species distributions at any scale. How complete are surveys, and how does the information decays with time and space? Why is this? Why we are looking at the decay? Because as you may know, things in space, the first law, geography, Euler's law, that although Wallace was already talking about similar things, is that things in space are similar, but they are similar the closer they are. Everything has been equal, we will expect more similar values at shorter distances, okay? So there is some information coming out of the records that stands throughout space, but that information decays with space. And the same happens with time. As information ages, it is progressively less and less useful to describe the current distribution of biodiversity. So we have formulated this in a very basic way. So the accuracy of the knowledge about a given taxon or community in a given area is a function of the knowledge about such taxon of community in that area in the moment of the surveys, the degree of completeness of the surveys and the quality of the records, and the loss of knowledge across time as it ages and across space, assuming that many records coming from other places are informing about the presence or absence of that species or about the structure of the community in that particular place. Okay, we have been working in the different aspects of this formula, of this basic formula. The first one is taxonomic uncertainty, thanks to the Taxon Time project led by Juliana Strop. This is an example. We have been trying to map and describe the uncertainty associated to the process of taxonomic description and synonymization. Okay, this is an example from the genus Atalia, one species, Atalia butyracea, that has, in Atalia, we have 148 accepted names. It's a palm out of 708 synonyms. And this is an example with one species that comes out about 18 heterotypic synonyms. An heterotypic synonyms is a type specimen which has been synonymized. Okay, we have 19 types, for that particular species. This looks quite clean and neat, but if we put together all the palms, this is what we get. We've got an entangled bank, okay? There are the palms, the acacia, have 1,091 names, almost 1,000 heterotypic synonyms, and only 175 species. And if you look here, there are some species that have been quite stable through time. Those are, we may be quite certain that those species won't change in the future, but there are also other species that have been changing continuously through time, being synonymized or reinstaurated many times. So we can assume that those species are more uncertain. We are trying to develop metrics that measure such uncertainty. We have also continued working on this data completeness and coverage, describing this to identify the spatial decay in information, assuming that the closer, not only the closer in space the data points are, the more information they are providing about the focal point or the point of each point of interest, but also that the more similar their environmental conditions are, the more informative they are. Okay, we have been mapping it, and we have quantitative ways of mapping that. Unidentifying, in this case, is the Iberian mosses. In another paper from Cristina Ronquillo, we are identifying areas with very little information, which are the clear areas that you see in the map in the lower down point. Also, we have been tracking how good is the information through time, and perhaps with no surprise, we identified that there were no places where we could track changes in communities throughout the whole of the last century. Okay, there we don't have good temporal coverage for any place in the Iberian Peninsula for mosses, which is a relatively well-studied group, at least compared with many arthropods and others. We are also trying to find ways of modeling this temporal decay and modeling how much information can we get through time from progressively older records, taking into account dispersal processes, demographic processes, ecological processes, and taxonomic changes. Okay, it's a work by Janet Sarolo. And we are putting together all this information into composite maps of uncertainty that take into account the information quality and how each focal cell is informed by other cells in space and by all the information from these cells and others in time. Okay, this, as you can imagine, this is quite difficult to parameterize, and it took us years to work it around, but we did it finally, and we published it a couple of years ago, almost 2 years ago, this paper in Ecography, where we produced the first maps of ignorance, of biogeographical ignorance, in this case with 3 dung beetle species in the Iberian Peninsula. And in the left, you've got maps of the observed occurrences, and the blue maps are predicted suitability, the results of species distribution maps. And the reddish maps, the orange maps, are maps of biogeographical ignorance, okay? Being 1, the higher ignorance, the less we know, or the less our data is informative on the distribution of that particular species, okay? And the clear areas, those areas where the data is good enough so as to provide us good, certain information about the presence or absence of that species. If we cross those 2 maps, we have this map of the prediction uncertainty, where, for example, you can see the yellow areas is areas where the model is predicting that the species is present, and we can be quite certain that the species is potentially present, okay? And in purple is areas where the model is predicting that the species is present, but we don't have information to corroborate that. So that prediction is quite uncertain. And the same happens with the absences. The gray areas are areas where we can be certain that the species can't live, and or at least according to the model we have. And the blue areas are those areas where the model is predicting that the species can live, but we don't have enough data, or the level of uncertainty about that response is too high. Okay, so what can we do with those maps? Okay, they are spatially explicit potential error terms. okay? They show the error associated to the data, not the whole error of the model, only the one associated to the biological data, okay? This can help us, of course, as we have seen, planning additional surveys in the areas where we have high uncertainty and are maybe important for conservation or other purposes, but also to highlight the uncertainty in the results of our species distribution models that we are using a lot in many places. And perhaps, and that's something that comes out of a paper of Colin Beale and Jack Lennon more than 10 years ago, is that we can incorporate such data uncertainty in the SDM as other techniques, incorporating those terms as error terms in regression-based models, or incorporating into Bayesian assessments, etc. If we put this together with information, with other sources of uncertainty, we will have a more fair account of all the uncertainty in the models that we are presenting to the public and that we are assessing between us. So let me conclude with this idea we've got of the whole process of integrating this data-driven uncertainty into the scenarios of biodiversity and ecosystem services. We have to work with heterogeneous data, so we have to first filter, identify the filters we've got, and be clear about why we use those filters and how many records they filter out, and also account for the quality of the records we use and the decay with space with time, and produce those maps of ignorance that we can use to engage with the stakeholders. If they can understand the map of suitability that tells them that for a particular species, this area may be quite good in the future or right now, depending if we are making projections to the future or not, or to different scenarios, they are going to be able to understand also the uncertainty shown also in maps in colors. So they can take the decision of putting some money or some effort in preserving some areas, more because we have more certain predictions than others that may seem better, but for which predictions seem quite uncertain. Okay, so to conclude, I have 3 main take-home messages. First one is that biodiversity data is by definition incomplete and inaccurate, and there is nothing we can do about that, but to continue working and accumulating data, okay? But we can also account for the limitations of the data. We can improve the data we use and filter out records that are not useful for the question we have, and we can describe the bias on coverage and measure the uncertainty they generate. And we can tackle that uncertainty. I haven't talked much about that, but some people like Rob Anderson and others have worked on that by reducing the extent of the conclusions or the analysis we make to the areas of high certainty, okay? Or to at least to incorporate some uncertainty into model building or in the interpretation. And just to conclude, I'm going to think I haven't done this alone. There are many other people, but let me mention those colleagues: Richard Leidel, J.C. Sarolo, Just Rob, Chris Ronquillo, Tiago Freitas, Tadeu Sobral, Jorge Lobo, Alexandre Diniz Fillon, Javier Nori, and many others. So thanks, and if you have questions, I'll be happy to answer them.